So in this video, we're basically going to be covering the whole of the carbohydrate syllabus and including tips on how to approach questions at the end. So we're going to be talking about the structure of alpha and beta glucose and the 5-carbon sugar ribose. We're going to be talking about how those monosaccharides bond to form disaccharides and those 3 disaccharides that you need to know about. And we're also going to be talking about the structure and function of polysaccharides. First, a little bit of context. And I've entitled this "You Are What You Eat" because here am I trying to make the connection between the biomolecules that you have to learn about at A level and our diet. Those groups of molecules that you will have learnt about in GCSE biology and in your food lessons. So it's a case of any consumer animal has to eat these molecules, they digest them, they then get turned into new carbohydrates, lipids and proteins in the consumer's body to make up the mass of that organism, the organism's biomass. So, quick family tree of carbohydrates. Carbohydrates made up of carbon, hydrogen and oxygen. That ATE at the end tells you that there is oxygen in the molecule. 2 main branches, sugars and complex carbohydrates. And sugars are sweet and they are small molecules. And complex carbohydrates aren't sweet and they are very much larger. And examples from your GCSE sugars, for example, glucose and fructose that you will have come across, and complex carbohydrates that you'll know about, starch and glycogen. So let's get started on the content, and we're going to start off with monosaccharides. Monosaccharides, mono meaning one, sac meaning to do with sugar. And these are monomers, small individual molecules that join together to make polymers. General formula for these are multiples of CH2O, where N can be between 3 and 7. And the 3 main groups that you are going to come across in A level are the triose sugars, those with 3 carbons, the pentose sugars, those with 5 carbons, and the hexose sugars with 6 carbons. And the 5-carbon sugar, an example is ribose, and examples of 6-carbon sugars would be your glucose and your fructose. So now we need to cover the structure of an alpha glucose molecule. So this is me drawing it out in full. It's a ring structure with an oxygen atom completing the ring. Each of the carbons has 4 bonds on it. And here's the really key part here, and I'm drawing it in yellow to show you the orientation of the hydroxyl groups in alpha glucose. Both hydroxyl groups are at the base of the molecule. And here's my very much simplified drawing of alpha glucose, again just showing where those hydroxyl groups at each side of the molecule are. And here am I numbering the carbon atoms, so that when we talk about bonds in a little bit, you know where those numbers come from. Each carbon is numbered so that we can talk about a 1,4 glycosidic bond, for example. So, in contrast, beta glucose, I'm just going to draw the simplified diagram for this. Notice that the hydroxyl groups are diagonally opposite each other in a beta glucose molecule, not both down at the bottom of the molecule in alpha glucose. It's a small but crucial difference. Properties of glucose, why is glucose so useful? Number 1, because it's soluble. and therefore it is easily transported. And number two, because the bonds that are contained within glucose contain a lot of energy, and therefore glucose can be used in respiration. However, it's worth you noting that only alpha glucose can actually be used in respiration. Beta glucose isn't. So now we're going to move on to ribose, which is a pentose sugar, 5-carbon sugar, because you also need to know the structure of this sugar. Again, it's a ring compound, the ring being completed by an oxygen molecule, each carbon having 4 bonds coming off it. And again, I'm going to number the carbons off 1, 2, 3, 4, 5. 4 of them in the ring structure and 1 outside of the ring. And the significance of this sugar is that it is used in RNA, ribonucleic acid, the cousin of DNA. So now we can move on to disaccharides. Di means 2, again sac means sugar, and this is where 2 monosaccharides are joined together with a glycosidic bond. Key term here, all carbohydrates, disaccharides, and as we're going to learn in a minute, polysaccharides are joined with glycosidic bonds, and glycose simply means carb related, to do with carbohydrates. You'll know it's a similar word as glycogen that you'd recognise from your GCSE. So how do 2 alpha glucose molecules join to make maltose? So here am I drawing my quick sketch alpha glucose molecules. Again, notice that the hydroxyl groups are on the bottom of the molecules, both of them. And you'll be able to see here when I draw the maltose molecule what has happened. Because instead of 2 OH groups on either side of the 2 alpha glucose molecules, we've now got a single oxygen molecule, which is the glycosidic bond holding the 2 alpha glucose molecules together now. And you'll notice that that's been caused by a water molecule being created from the 2 alpha glucose molecules. This is called a condensation reaction because water is produced. In contrast, the reverse reaction is a hydrolysis reaction because water is used to split the maltose into the 2 alpha glucoses. Hydrolysis meaning water splitting, or splitting with water. And in the same way, polysaccharides are formed because simply more monomers are joined onto either side of that glucose molecule, that maltose molecule, to make a polysaccharide. So the 3 disaccharides that you need to know about and what their constituents are maltose made from 2 alpha glucose, sucrose made from an alpha glucose and a fructose. Notice that sucrose is the sugar that you have on your table at home, and fructose, that fruit sugar that you learnt about at GCSE, which is really sweet, is what makes that table sugar kind of particularly sweet. And the final one is lactose, which is glucose and galactose. Now, jumping back to our carbohydrate family tree, I just want to point out that sugars are mono and disaccharides, and complex carbohydrates are polysaccharides. So we are now able to move on to polysaccharides. Remember, these are those complex carbohydrates, large molecules, not sweet. These are not sugars. And these are all polymers, as the word poly suggests, poly meaning many. And there's your definition of a polymer. It's a large molecule made up of many monomers. And these are the 3 polysaccharides that are covered by A-level syllabuses, except Edexcel does not include cellulose. So, first of all, starch. Crucially, you need to know what the overall function is. It is the alpha-glucose storage molecule for plants. You will never find starch in an animal cell. Really important that you try and remember that. And this starch substance is actually made up of 2 polysaccharides. Amylose, which is the one that AQA focuses on, but also, in addition, a amylopectin. And if you are doing OCR or Edexcel, you absolutely need to know about amylopectin. So starting with amylose, here am I trying to draw a simple diagram of amylose. At the start, including the monomers of alpha-glucose, and continuing the diagram without them, it's a helix of alpha-glucose monomers. And the helix that is formed from those alpha-glucose monomers is stabilised by hydrogen bonds, which form within the molecule. So amylose is an unbranched polysaccharide, and it's made up of alpha-glucose molecules. And these alpha-glucose molecules are bonded by 1,4 glycosidic bonds. This is not a detail that AQA focuses on, but the other 2 exam boards do. So that is the crucial structure of amylose. So how does the structure relate to its function? So remember, the overall function is alpha-glucose storage. Number 1, it is insoluble. It's a large molecule, and large molecules often are insoluble. And importantly, that means that it doesn't affect the water potential of the cell. Those 3 dots means therefore. It is also compact. Therefore, lots of glucose can be stored in a small space. And finally, it's made of alpha-glucose, and therefore it's used for respiration. These are 3 properties and 3 explanations of those properties. Notice that from an exam question point of view. Amylopectin. Here am I drawing a simple diagram of amylopectin, starting off with the individual monomers being drawn, and then I'm going to continue the diagram without that detail. So there's your alpha-glucose monomers. And this clearly is different from amylose because it is a branch molecule. And it contains alpha-glucose, which are bonded by both 1,4 and 1,6 glycosidic bonds. AQA students don't need to worry about that detail. And again, now let's link the structure and the properties related to the function, and you will notice that the same things come up again. Notice you've got the structural or the property, the structure or the property, and right next to it you've got an explanation as to why it is useful to the overall function of the molecule. Exactly the same as above, except for here we're going to add in the fact that this molecule is branched, and that means that there are more ends that can be hydrolyzed, so that glucose can be released, can be hydrolyzed faster. Now that word faster is important. In mark schemes, easily will not get you the mark. Faster is required. And finally, we're going to move on to glycogen. And here's the overall function of glycogen. It is the alpha glucose storage molecule. And here's the important bit for animals. Glycogen is not found in plants. And I haven't bothered drawing in the individual monomers on this diagram, but I'm really hoping that you're starting to realise what the difference is between amylopectin and glycogen just from this diagram. Because clearly there are similarities. They are both branched molecules, except that obviously glycogen is very much more branched than amylopectin. And again, 1,4 and 1,6 glycosidic bonds are used between the alpha glucose molecules. And here are the key points again of how the structure or the property of the molecule relates to its function. And there are repetitions, but I'm doing this deliberately because these are the structures and the properties that are talked about in mark schemes with the accompanying explanations. Insoluble, so that it doesn't affect the water potential of the cell. Compact, so that a lot of glucose can be stored in a small space. Alpha glucose being used so that it can be used in respiration. But now I'm really going to try and emphasize why glycogen is so important for animals. And it's because it is very highly branched, and therefore there are very many ends which will allow glucose to be hydrolyzed off the molecule to be used in respiration. And again, important that you use the word fast hydrolysis, not easy hydrolysis. Easy does not get you the mark. And the reason why this is particularly suitable for animals is because they have both a higher metabolic rate and a more varied metabolic rate than plants. Let's face it, animals move more quickly than plants, and they can go from rest to full sprint in a matter of seconds. So that fast hydrolysis of glucose is important. So finally, we're moving on to cellulose, which has a very different overall function to both starch and glycogen. And that's because this polysaccharide, this polymer, strengthens plant cell walls. It's a strength polymer, not an energy polymer. And that's because it is made up of beta-glucose. And as I'm about to show you, alternate beta-glucose monomers are at 180 degrees to each other in the polymer of cellulose. So here are my beta-glucose monomers with the hydroxyl groups diagonally to each other, and notice that the second beta-glucose monomer has to flip through 180 degrees for those two hydroxyl groups to be close to each other so that the condensation reaction can occur. 1,4 glycosidic bonds are used. But notice, because alternate beta-glucose monomers are at 180 degrees to each other, this forms a straight chain molecule. It's not helical and it is not branched. So here am I trying to draw those glycosidic bonds kind of alternately at the top and the bottom of the molecule as it goes along. And this is also important because the cellulose molecules are straight chains. They line up next to each other and therefore form hydrogen bonds between the cellulose molecules. And when a bunch of cellulose molecules are joined together through many hydrogen bonds, we call that a microfibril. So here are my glycosidic bonds being drawn, and now I'm putting in the hydrogen bonds. So I've drawn here a very small microfibril. Notice that the hydrogen bonds are between the cellulose molecules, not within them. Individually, hydrogen bonds are weak. You would never call a hydrogen bond strong, but many of them in a molecule confer or give great strength and stability to the molecule. So let's now summarise how the structure or the properties of cellulose relate to its function, its primary function being strength. And it all stems from the fact that beta-glucose has been used. Beta-glucose is used, which means that straight chain molecules are formed, which means that many hydrogen bonds can form between those molecules, and therefore, as a result, a strong cell wall results. If you had to pick one of those things, the strength would be as a result of the many hydrogen bonds. But you can see that it flows sequentially as a result of the beta-glucose molecule being used as the monomer. So, questions that come up. You need to know the content of this video. You might be asked to describe the molecules. Pretty straightforward, hopefully, if you know the content. If you're asked to compare different molecules, notice that you have to compare the same feature in both molecules. This is often not done by students and therefore marks are lost. So, for example, with amylose and cellulose, amylose use alpha glucose, cellulose use beta glucose. I'm comparing the monomer that is used. And in addition, amylose has hydrogen bonds within the molecule, whereas cellulose has hydrogen bonds between cellulose molecules. So I'm comparing the same feature. Another type of question might be to explain how structure is related to its function. This is why I have laid out the structure and function parts of this video the way I have. I have lifted them from the mark scheme. So I've named the property and I have explained why it helps the overall function of the molecule. Here's a slightly higher version of an explanation, and that is why glycogen is more suited for animals than a amylopectin. And that's because glycogen, because of its highly branched form, allows much faster hydrolysis in animals which have a higher metabolic rate. And finally, you might be asked to apply, you might be given the diagram of a novel monomer that you've never come across, and you might be asked to suggest what kind of polymer it forms. And my advice would be is to look at the orientation of the hydroxyl groups, whether it's like alpha glucose or beta glucose, and then go from there as to what kind of polymer it would form. I hope this has been useful. I'm Biology Carol. If it has been useful, please like and share, and do leave comments below. I want to make these videos better as I go along.